Hey, it's Mr. Lum. Sorry I can't be there. But I'm going to be teaching you the genetics portion of this course. And so we're just going to start off with a little bit of review from what we remember from grade 9. So do you remember what is the difference between mitosis and meiosis? You can feel free to put the video on pause so you can have like a discussion of maybe some of the things that you think. But hopefully you came up with a few things like this. Both of them have PMAT, or prophase, metaphase, anaphase, and telophase. The only thing is that mitosis does PMAT once and meiosis does PMAT twice. And thankfully it's just called prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. If we remember what crossover is when you have one gene that you have from your mom, one gene that you have from your dad. They line up during meiosis, and then they switch some genetic information like this. And they kind of mix and match chunks of genetic information, and that just ensures that meiosis, if you remember what meiosis is, that is making sperm and egg. It's used for sexual reproduction, but crossover only happens in meiosis, and it just ensures that every single person is different. So if hypothetically, if a mom and a dad happen to have 1,000,000 children, which is impossible, but if they did happen to have 1,000,000 children, each one of those kids would be genetically different. Not completely genetically different. They would look like all siblings and everything, but they would look a little bit different because of crossover. Another thing too is they would look different because of something called independent assortment. So during metaphase, if you remember, independent assortment is when those chromosomes in metaphase 1 line up like this, and they get pulled apart to separate sides. That the way they line up kind of randomly. So instead of writing X's, how would I say something like this? Chromosome 1 is here, 2, 3, 4, 5, 6, 7, 8, and then obviously those would get separated into 1, 3, 5, and 7 on that side, 2, 4, 6, and 8 on that side. But what happens if they lined up differently? Okay, and let's, instead of lining up like that, they lined up like this. So this would turn into one sperm cell and another sperm cell of 1, 3, 5, 7, and 2, 4, 6, 8. But if on a different sperm cell, let's imagine they lined up like this: 1, 2, 4, 3, 5, 6, and like that. They're still lined up across from their proper pair, but what would happen is that this one would be 1, 4, 5, 8, and this sperm cell would turn into 2, 3, 6, 7. So this sperm cell, all coming from the same parent, would have chromosomes 1, 4, 5, and 8. This sperm cell would have 2, 3, 6, and 7. This one would have 1, 3, 5, and 7, and this one would have 2, 4, 6, and 8. And so that's called independent assortment. That's just another measure to make sure that every single sperm cell is different, every single egg cell is different. So, do we remember what these words mean? 2N. 2N means it has the full set of chromosomes, and this means has the half set of chromosomes. What's a full set of chromosomes for a human being? Is 46 chromosomes. Half set is 23. What cells have a half set of chromosome is our sperm and egg cells. So sperm cells only have 23 chromosomes, egg cells only have 23 chromosomes. A normal human has 46 chromosomes. We typically call this haploid, and we typically call this diploid. Okay? So, if we remember in prophase, metaphase, anaphase, telophase, prophase, the nuclear membrane starts to disappear. Metaphase, they line up in the middle. Okay, anaphase, they A for away or A for apart. The chromosomes get pulled apart. And telophase is the nuclear membrane starts to reform around the DNA, and the chromosomes start to turn back into their DNA long strands, and it's starting to turn a little bit back into two cells. So if I were to draw a picture of it, prophase normally looks like this, and normally you have DNA in there, and that DNA starts to turn into these X-shaped chromosomes, and that's an early prophase and a late prophase. That nuclear membrane starts to disappear. So I'm going to draw, here's my nuclear membrane starting to disappear. We got those X shapes in there. By metaphase, the nuclear membrane is completely disappeared, and all those chromosomes line up in the middle. And in anaphase, those things get pulled apart by spindle fibers. And then telophase, start to get that weird peanut shaped. And then you have all those sister chromatids right there. The nuclear membrane starts to reform. So this is drawing some nuclear membrane going back around this stuff and starting to reform again. And that's our nuclear membrane starting to reform, and that's our telophase. Now, in meiosis, it does that as well, but it just does it twice. It does PMAT once and then PMAT a second time. Okay? And a few other little changes. So one change is that prophase is pretty much the exact same thing. Chromosomes form up, but the only difference is that crossing over occurs. We talked a little bit about what crossing over is. And then metaphase, one is it lines up in the middle again. But the difference between meiosis is that now in meiosis for metaphase 1, instead of getting that single line of chromosomes, those homologous pairs, that means you get one from your mom and the other one you get from your dad, they line up in the middle. Okay? And that is, this one right here is metaphase 1. And we talked about what independent assortment is. They line up randomly. Now in anaphase 1, instead of chromatids getting pulled apart, the homologous pairs get pulled apart. So in anaphase 1, these things get pulled apart. And then telophase 1, same thing. The nuclear membrane goes around them, peanut shaped still. And then we have, instead of chromatids, we have whole chromosomes. And then the nuclear membrane starts to form around it again. And that is telophase 1. Now prophase, metaphase, anaphase, telophase 2 is the exact same thing, and it looks very similar to mitosis. In metaphase 2, we get that single line of chromosomes. So we're back to the single line of chromosomes. And then the chromatids get pulled apart. So just like mitosis, chromatids get pulled apart. And just like mitosis for telophase 2, that peanut-shaped nuclear membrane forms, and we have all those half chromosomes, which are also called sister chromatids. form in there, just like that. Okay, now these have half the number of chromosomes, so they're called 1N, or in other words, they are called haploid. Okay, and when they have the full number, that 46 chromosomes, it's called diploid. Now, haploid cells are only reserved for sperm and egg. Now, these haploid cells are for gametes or sex cells, sperm and egg. Now I want to emphasize, like, us humans have 23 pairs of chromosomes, or 46 chromosomes in total. The other animals will have different number of chromosomes. I can't remember what it is. I think a cow is like 60 chromosomes. A fruit fly is like 10 chromosomes. The number of chromosomes actually doesn't matter. Like, there's no relationship between the size of the organism and the amount of chromosomes. It's not like a cow is bigger, it needs more chromosomes. A fly is smaller, it needs less. It's just the way the information is packed. But different animals have different number of chromosomes. Some animals have the same number of chromosomes, especially if they're more closely related. But for the most part, different number of chromosomes. But here's a diploid cell, which is like a body cell, and it has, here's the first pair, or 2, and then here's number 22, and then the last pair right here. These are called the sex chromosomes. And then the haploid, here it just has the 1 chromosome, so 23 chromosomes. Okay. Now on our other piece of guided notes, I don't have this part down, but on your other piece of guided notes, it says DNA stands for, and it stands for deoxyribonucleic acid. And DNA is made up of nucleotides. And it says a nucleotide is made up of 3 parts, and I don't have that here, but we're going to talk about that. They are made up of, part number 1 is they're made up of sugar. Now, it's not the type of sugar that you're used to. So in chemistry, sugars are things that end with ose. So you may have heard of glucose, fructose, sucrose, maltose. Anything that ends with ose in chemistry is a sugar. Now, the type of sugar that is in DNA is called ribose. Now, so that's part number 1. And the next part is phosphate. PO4. And then the final part is a base. Now the base is the most important type. These are the 4 nucleotide bases: adenine, cytosine, guanine, and thymine. Now, these nucleotide bases are, adenine will only combine with thymine, and guanine will only combine with cytosine. And DNA is in this, what we call a double helix formation. It's like this ladder formation that has been twisted. And let's see, these parts right here, these are the sugar and phosphate and sugar and phosphate, and here's the more sugar and phosphate and more sugar and phosphate. This outside parts are the sugars and phosphates. Now where's the bases? The bases are these parts right in here. these are the bases. They connect one side of this twisted ladder to the other side of the twisted ladder. They connect them using what's called hydrogen bonds, which you don't really need to know about it. So it's not like a chemical bond, it's just like an attractive force that connects one side to the other side. But this is where right in the middle part is where the bases are, and on the outside part is the sugar and phosphate. So again, here's another picture of it. Here's that sugar phosphate on one side, here's the sugar and phosphate on the other side, and you can see A only combines with T, or adenine only combines with thymine, so only yellow and green will ever go together, and cytosine and guanine will always combine. In other words, blue and red will always go together. Okay. Now, DNA is the blueprint in our body, and it makes up everything, and therefore DNA is very, very, very long, and it needs to be packed into the body in a very special way. And DNA from just one cell will be approximately 6 feet long. And how can 6 feet long pack into one cell? Well, it needs to pack into a very specific way. So DNA is twisted and rolled into what is called chromatin, and chromatin can be twisted into an X-shaped structure called a chromosome. Now, here is a picture of like how DNA is packed. So here we have our DNA. Okay, again, adenine combines with thymine, adenine with thymine, thymine with adenine, cytosine and guanine. Okay, and so that is all packed up in here. And then what you have is like a protein called histones. And I like to think of it as histones are like toilet paper rolls, and the DNA is like the toilet paper. Now, DNA will pack a lot tighter if it's going around these toilet paper rolls, and we can pack DNA to a much smaller kind of thing. So think of it like toilet paper. If you unrolled it, it would take up a lot of space. But since you have the toilet paper roll, it is able to roll around the toilet paper roll and pack up into a much tighter space. So these histones allow it to pack up in a tighter space. And those histones then coil up and get even more compacted into chromatin. And chromatin then can get compacted even further, and that makes what is called a chromosome. And that is how it kind of gets packed in there. Now, at this point, you can put the video on pause and watch a video of how histones come into play for packing DNA, and then we can come back to this video here. So the next thing is we talked about this already. Humans have 46 chromosomes, and there are 23 pairs of chromosomes, and each look very similar except for the last one. The last pair right here look different, and these are what we call the sex chromosomes, and they look very, very different. Sex chromosomes don't really exchange too much DNA. I shouldn't say that. Sex chromosomes don't exchange DNA for males. So this is a human male, which has an X chromosome and a Y chromosome, and the two chromosomes look very, very different. The X chromosome has a lot of genetic information. The Y chromosome has little to very little genetic information on there. And we'll talk about the significance of that later. It means that males will typically be more prone to different genetic disorders or say certain genetic diseases than females. But we'll talk a little bit more about how that works. A couple definitions that we're going to be using quite a bit are the term genetics. And genetics is the study of how inheritable characteristics such as eye color or in humans, or flower color in plants, are passed from one generation to the other. And this is all done by genes. A gene is a section of a chromosome that codes for, or is the blueprint to make one specific protein in a body. So there'll be one gene on a chromosome that'll code for your eye color, one gene that might code for hair color. And yeah. And each one of those genes creates a protein. And we're going to talk just very briefly, because you should have talked about it in grade 9, of how a protein is made. So a protein is made when, first off, a gene is copied by, this should say a gene is copied into messenger RNA. Okay, so we have our main piece of DNA, and then a small chunk of that DNA, so maybe say this chunk of DNA is copied, and we call it messenger RNA. There's a few little differences between RNA and DNA, but they're quite a bit similar. Now, after that messenger RNA then leaves the nucleus, that messenger RNA then leaves the nucleus, that messenger RNA then goes to a ribosome, and the ribosome is like on the endoplasmic reticulum, or it is or somewhere else in the cytoplasm. That ribosome, which is right here, takes in the messenger RNA and reads it. And when it reads it, it then translates it and brings in amino acids. Amino acids are the building block of DNA. And when it brings in those amino acids, it combines hundreds and thousands of amino acids and turns it into a protein. Now, this whole process right at the beginning part, where it goes from DNA into RNA, that is called transcription. Okay, so the DNA is rewritten into RNA, or in other words, it's transcribed into RNA. And then that RNA is then translated, or translation, into what is called a protein. So there's two parts in making a protein. There's transcription, and transcription is essentially going from DNA into messenger RNA. That's what transcription is. What's translation? Translation is when we have messenger RNA is read and turned into a protein. Now, hopefully now you're going to be watching a video on how a protein is made, and hopefully you'll be able to see transcription and translation.